So it's really a terrific pleasure for me to introduce one of my favorite people to you, Mete Civelek. Is that the right pronunciation? And he is originally from Turkey. And one of the weird things that I read about Turkey, I read a lot about Turkey because of family connections, is that it ranks low in the percent of the population that believes in evolution. I don't know why, but it's like one of the lowest. But Mete represents a really fine specimen of human evolution. Because he started out just straight up strict bioengineering with a really strong interest in flow dynamics. And he went to the University of Pennsylvania where he earned his PhD degree studying flow dynamics in the vasculature. And that was, that was his focus. But then he went to the lab of Jake Lucis at UCLA, another one of my favorite people, who is really broad. He's all over the place, but he's a fantastic geneticist. Mostly mouse, but he does a lot of human genetics too. And he evolved into a geneticist. And he's a fantastic geneticist. He's doing so much wonderful work in a whole array of areas. At Jake Lab, he did quite a lot in mouse genetics, but he also developed his interest in human genetics, and he studies a lot, I'd say more than anything, on atherosclerosis, but not exclusively. He also studies the genetics of obesity and diabetes. And he does really very, very exciting work using cells of the arterial wall, smooth muscle cells, endothelial cells, macrophages, studying them ex vivo and doing a lot of very nice molecular work on cis and trans eQTLs and networks of gene expression and all of that from the ex vivo models that he can then use to understand the genetics of atherosclerosis. Really elegant work, and I'm so happy to have him here and met him.
Thank you so much. Thank you. I hope, can everybody hear me in the back? Okay, perfect. Thank you so much for that kind evolutionary introduction. It's really true. I evolved. I hope you guys know my colleague, Brian Parks here, also my friend. So we used to sit right next to each other in Jake's lab, which is a genetics lab, and none of us come from a genetics background. And we would say to each other, like, what is this genetics, dude? Like, this makes no sense. Like, I don't even know what they're studying, what is an eQTL? It took us probably 6 months to a year to even understand the concept of eQTLs. So at the end of 5 years, we're like, oh my God, genetics is amazing. We're going to devote our lives to genetics, basically. So, we bit the genetics bug and we've been doing genetics since then. And that's what I've been doing in my lab. And I'm very happy to share that today. My lab studies coronary artery disease, which is the, leading cause of death in the United States. And it certainly has an environmental component, right? Our lifestyle plays an important role in the risk, smoking, exercise, diet, etc. But it also has a pretty significant genetic component. The heritability estimates from, twin studies, family studies is, anywhere between 40 to 70%, indicating a strong genetic component. So in the last 15 years or so, we've been doing genome-wide association studies to identify the genetic component of the coronary artery disease. This is a Manhattan plot, which I'm assuming this is a genetics department. Most of you are very familiar with these. From the most recent GWAS of coronary artery disease that included about 1.1 or 1.2 million people, and identified 320 loci that are associated with the disease risk. And of course, it is very impressive, right? You phenotype this many people, you genotype this many people, you bring a lot of cohorts together, you replicate these studies, and you find these loci, and it's almost the beginning, right? We need to now interpret these loci to understand more about the disease biology. when we look more closely into these loci, we realize that some of them actually are also associated with well-known risk factors like lipids, like obesity, like vascular function, etc. But majority of the loci have unknown function. So about 60 to 65% of the loci have unknown function. And even if, they're annotated with a certain gene that's maybe nearby, that plays a role in, transcriptional regulation, etc., you still don't know, what is the regulatory element, which cells, which tissues, what it's regulating, etc. So there's a lot of unknowns. The underlying cause of this coronary artery disease is a process called atherosclerosis. And, I don't know how many of you study cardiovascular disease, so I wanted to give you just a very brief introduction to atherosclerosis. It's essentially the buildup of the, plaque in the artery. So this is a healthy artery that has these three layers, the endothelial layer, the medial layer, which has smooth muscle cells, and the adventitial layer that has the fibroblasts. And with time, due to lipid accumulation, macrophage accumulation, etc., you get these plaque buildup. And the plaques can be in one of two states. They can have this thick fibrous cap, which are, proposed to be stable plaques, but they can have these thin fibrous caps, which can rupture, and then those can lead to heart attacks and stroke. So this is the underlying disease process for coronary artery disease. When I started my lab, we had a very simple hypothesis. So in the disease process, the smooth muscle cells that are in this medial layer that are normally contracting and relaxing and adjusting your blood pressure moves to this subintimal space, and they change their phenotype. They become more migratory, proliferative, and really in advanced stages, they actually calcify. And, there's now a lot more single cell studies that have been published, and we have more granular understanding of this process. So it's not like the cells go from just a quiescent state to an immediate proliferative state. There's obviously steps in between, and single cell studies have kind of mapped those transitions. But when I started the lab, we thought, well, this is a disease that develops in the vascular wall. So some of the variants that are associated with the disease should be affecting the vascular function, at least a subset of them, right? And obviously that was an extremely simple hypothesis that we decided to test. So our goal then became to do this mapping from, the variant in the locus to its effector gene, and then try to figure out what the, gene does, what's its function in the vascular wall. And we have actually two translational goals, big goals with this. One of them is, identifying the causal variants in these loci to help with these polygenic risk score predictions. So currently, you can use these association data sets and calculate polygenic risk score for each individual based on their SNP configurations and the effect size of these SNPs. And, it's basically then gives you an overall genetic risk score for each individual. But these polygenic risk scores, if you, for example, identify them in people with European ancestry, it doesn't translate well to people with Asian ancestry or African ancestry, etc. And it's because we use essentially almost all the genotype SNPs, and their SNPs are highly correlated to calculate these polygenic risk scores. So if we actually find the causal variants in each of the loci, we will have a lot less noise and a lot more confidence in our predictions for polygenic risk scores. So that's at the SNP level. The second one is if we can identify the causal genes, or the effector transcripts as it's called, we actually improve our chances of finding drug targets that can be efficacious. So there are many studies, this is just one of them, where people looked at, the drug targets that pharmaceutical companies tried to target for various diseases, and if they failed in phase 1 or phase 2 or phase 3 clinical trials. And basically, the targets that had a GWAS evidence were twice more likely to go through the process of, phase 1, 2, 3 clinical trials successfully and get FDA approval compared to those that don't have, any GWAS evidence. So this basically was, our overall motivating factor to have a translational impact by looking at the SNPs, finding the SNPs and the effector transcripts. Okay, so how do we identify the causal variants in the loci? So this is the first question that I'm going to try to answer. It turns out that this is not a trivial problem. This is a zoomed in version of the Manhattan plots. We call these locus zoom plots. Each of these dots are against SNPs. And this is the association, the minus log 10p, and this is the location of the SNPs. And what you can notice is that there's a group of SNPs that are really highly correlated because they're in high linkage disequilibrium. So it's not easy to just looking at this plot say this is the causal variant in this locus. The second thing is they're in mostly non-coding regions. We actually did a study and found that less than 1% of all the SNPs that are in coronary artery disease loci lead to amino acid changes. They're in coding regions. So 99% of them are in non-coding regions, and majority of them happen to be in enhancers in various cell types. And the third question is obviously, cell type specific. Are they acting in endothelial cells, smooth muscle cells, liver cells, adipocytes, etc. So these are the kinds of questions that we're going to tackle. And how do we do that? The way my lab thinks about this is, let's basically integrate a lot of different approaches to identify some of these causal variants. So this is obviously a very simplified version of a gene regulation, locus, right? You have the promoter, you have the enhancer, you have these histone marks, you have the target gene. You can imagine a variant, for example, that affects the binding of a transcription factor, and we can actually assay that with these parallel reporter assays. We can look at epigenetic marks using epigenetic marks and a TAC-seq to basically find open chromatin regions where these variants are presumably there and acting to regulate gene expression. And then we can do EQTL mapping to figure out the target gene. So basically, throughout this talk, I'm going to give you examples from all of these things that we did in the vascular wall cells. All right, so we need to start with the SNPs now. So we took basically the SNPs in the GWAS locus and actually used a pretty permissive linkage threshold, and we looked at the 4 major populations from the 1,000 genomes, and we ended up with essentially 26,000 SNPs to query. So now we're going to look at which of these SNPs have a gene regulatory function. Now, this is a pretty large number of SNPs to actually assay. So typically what most people do is they do some kind of fine mapping. They look at the SNPs in regulatory regions, open chromatin regions and things like that to narrow down this list. But we actually intentionally did not do that. And one of the reasons is some of these SNPs are also in loci that affect lipid levels or obesity. And we thought that they actually serve as a perfect biological control because we're going to perform our studies in vascular wall cells, and we should not see those SNPs that are associated with, non-vascular wall related processes in our data set, right? So that's why we use essentially all of these SNPs in our queries. So we add scramble controls, to this SNP, and each SNP obviously have 2 different alleles. So we have about 52,000 different sequences to test. And the way we do that is we synthesize these inserts, 200 nucleotide long regulatory elements, or just oligos basically, where the SNP is right in the middle. So you have one allele or the other allele in these oligos. And we clone them into a lentiviral vector that has a minimal promoter that's driving a barcode and a GFP expression. So we're really testing the enhancer function of these SNPs. And basically what we're looking for is there a difference between allele 1 or allele 2 of each of these SNPs in its enhancer function? And because this is a random process, basically per each sequence, we actually assay it on average 65 times. So because there are 65 unique barcodes that get inserted, in this process. Because once we create this library, we have to sequence it and assign the regulatory element to the barcode. So we get pretty, robust data basically because we're testing each SNP multiple tens of times. We are interested in the smooth muscle cells because we thought that some of these SNPs are acting in the smooth muscle cells to regulate gene expression. So basically, we take these lentiviral particles and infect smooth muscle cells from 6 different donors, 3 males and 3 females. But we also cultured them in the presence or absence of FBS because this is a pretty well actually known culture condition in vascular smooth muscle cells. In the absence of FBS, they become more quiescent and more contractile, kind of simulating, the healthy vascular condition, whereas in the presence of FBS, they're much more proliferative and kind of simulating the disease condition. So we're trying to test, how sex and this phenotypic state of the cells affect, gene regulation by these SNPs. So once you infect, you wait for a few days, and basically you extract the DNA and the RNA, because obviously the lentivirus inserts itself into the chromosome, and it's a completely random process. So you might get more than one insertion. So we're trying to basically count, how many of these sequences have been inserted, and the RNA that they are leading to the expression. And then all we're doing is really just calculating the ratio between RNA and DNA molecules as a way to look at the expression and how the SNPs are affecting the expression. So the first thing we did was we thought that obviously not all of these SNPs are going to have some kind of an enhancer or regulatory activity, right? So we compared the expression to the scramble controls. This is a density plot of the MPRA, this MPRA activity score. And basically what we found is that about 6% of these SNPs actually show an enhancer activity, right? So we were able to essentially narrow down our list from 26,000 SNPs to only 6% of those SNPs. And the next thing now we're going to look at is then allelic imbalance in the activity, right? Because that implies that the SNP is having an effect on gene expression. So we're looking for cases like this, where one allele of the SNP has higher or lower expression. And this is, of course, among the SNPs that already show an enhancer activity. So when we do that, this is I guess a volcano plot that show both enhancer activity and allelic imbalance. We actually end up with about 130, 140 SNPs that show both an enhancer activity and an allelic skew. And what is interesting, and again, I don't want to, infer that, these are obviously representative of the disease that happens in vivo, but there is definitely a phenotype-specific expression, right? So there are 70 SNPs that show this allelic activity or allelic imbalance only in the proliferative smooth muscle cells, and about 30 or 40 of them in the quiescent state, right? And there are some that are active in both cases. So maybe, right, maybe these SNPs are acting in different stages of the disease. And again, I don't want to draw too many conclusions because obviously this is in vitro. Okay, so now we have this 130 or so, 136 different SNPs. You remember I told you that, we want to make sure that these are the ones that are acting in the vessel wall. And it turns out that 30% of the SNPs that we tested were in lipid loci, but only 4% of these SNPs are in the lipid loci. So we definitely depleted, if you will, the search space, and we're focusing on the non-lipid associated SNPs, which we already thought were acting in the vascular wall. Okay, so now this is nice, but it's this kind of artificial assay, right? You're finding all these allelic activity, and the sequence is inserted into the chromosome, but it is obviously not the endogenous locus, right? So in order to look at the endogenous locus, then we turn to different epigenetic marks in the exact same cells, and we looked for SNPs that are showing these activity differences in the mPRA assays, but also are showing that are in open accessible regions or enhancers based on the epigenetic marks. Okay, so when we do that, so this is just one example. For example, this RS165 SNP, as you can see, it is in this accessible region in the proliferative state or the quiescent state based on all of these epigenetic marks. We actually now narrow down our search space to about 30 SNPs. So we think these 30 SNPs are potentially causal and regulating gene expression in vascular smooth muscle cells. But what we haven't done is actually connecting these cells, or these SNPs, I'm sorry, to gene expression yet, right? So how do we do that? How do we connect the SNPs to their target transcripts? In order to do that, we turned into this resource that was actually developed in UCLA, and that is this resource of vascular smooth muscle cells from the healthy heart transplant donors. So these are basically people who, unfortunately passed away in primarily car accidents or motorcycle accidents, and their families donated the hearts for transplants. And surgeons, when they do this procedure, they actually cut the aortic pieces so that they can, fit it into the recipient. And they would normally discard these ascending aortas. So they were taken, and then endothelial cells and smooth muscle cells were extracted from these individuals. And majority of these donors are male, because that is essentially the representative of the donor pool in the United States. And actually around the world as well, majority of the heart transplant donors are male. So now we have these cells, these smooth muscle cells from 151 different donors. What do we do with them? I had a postdoc, Redwan Rahru, and he had an army of undergraduates, because he did this really extensive characterization experiment. He took these cells, grew them, in plates, and measured 3 phenotypes that are relevant to the development of atherosclerosis: migration, proliferation, and calcification. And he did this in different conditions. So, for example, PDGF is a well-known chemotactic agent for smooth muscle cells. So he looked at, migration under both control conditions or PDGF conditions. These 3 cytokines, PDGF, TGF, and IL-1, are well-known stimulants of smooth muscle cell proliferation. So he did it under these different conditions. Similarly, for calcification, he used an increased inorganic phosphate media or an osteogenic media to look at these different aspects of vascular calcification. So basically, he did this extensive cell characterization, and we have several phenotypes of cellular phenotypes that are relevant to atherosclerosis from this data set. Additionally, he also did molecular characterization. He looked at accessible chromatin using ATAC-seq, and again, in these 2 different conditions, proliferative or quiescent smooth muscle cells. He looked at gene expression with RNA-seq, again, these 2 conditions. And then he looked at secreted extracellular matrix protein abundance from these cells. And the main reason is because, if you remember, I told you about the thick and the thin fibrous cap that determine the stability of the atherosclerotic plaque, and that's because smooth muscle cells secrete extracellular matrix proteins that kind of helps with that process. And he used to call this thing is GWAS in a dish because he's doing all these extensive characterizations. So I'm gonna not show you every single thing from this, and some of it is already published, so most of the stuff is, that I'm gonna talk about is unpublished work. So I'm gonna show you just some aspects of this. So the very first thing that we did was EQTL mapping. So we're looking for basically cases where a genotype of a SNP has an impact on the gene expression, right? So we're looking for cases like this where there's an A allele that leads to low gene expression compared to C allele where there's, a high gene expression. So we measured the expression level of 18,000 genes, and we had information from 6.3 million SNPs. So you do this linear regression, and you find basically 6,500 genes where there's a cis EQTL. So there's a variant near the transcription site of the gene that affects or that's associated with its expression levels. So once we have that, now we can connect this to coronary artery disease loci by doing this analysis called colloquialization analysis. This is just one example of a locus. So this is basically the coronary artery disease GWAS in this locus zoom plots in chromosome 7, just one locus. There's a nearby gene whose EQTL also maps to that locus, and you can just see how similar they look, in terms of the genetic signal. And when you actually align them, you see that they align across the, across this diagonal, which indicates that the same genetic variant is associated with both disease and also the gene expression. So overall, we had 85 genes whose expression also was associated with coronary artery disease variants. And so this is kind of an ugly plot, I'm sorry, but this is basically the location of the coronary artery disease SNPs and the genes that map to it, basically, starting with chromosome 1 all the way to chromosome 22. And these are shown with respect to the risk alleles. So the genes at the top have high expression in the presence of the risk allele, and the genes at the bottom have low expression in the presence of the risk allele. There are some genes that have been shown to play a role in atherosclerosis, but there are quite a few where we don't know what they're doing. And there are quite a few, for example, non-coding RNAs here, etc. And this became kind of our search space now to connect the SNPs to the genes, right? So the way to do this, to connect the predicted causal variants to the gene expression, is obviously by putting together all these evidences, right? As I stated, we're looking for orthogonal evidence. So we look for allelic imbalance in the MPRAs, the EQTL SNPs, the SNPs in accessible chromatin region, and we actually end up with 15 SNPs that we have multiple layers of information in these loci that we think are playing a role in the coronary artery disease loci. And this is the list. And again, this is not the prettiest way to show these things. And there are some interesting things. For example, there are SNPs which are associated with, more than one gene in a given locus, or there are more than one SNP that is associated with the expression of a single gene. So, it's a pretty probably complicated gene regulatory effect that's happening in these loci. So we have basically 18 SNP gene pairs. And the nice thing about this were actually 2 of these SNP gene pairs had already been shown in the literature to have an effect, and that these pairs, that this SNP actually regulated this gene expression. So we were pretty happy to see that we were kind of rediscovering at least some of the known loci, and gave us confidence for the remaining loci. Now, one of the things, again, we're doing here is, looking for evidence from all these orthogonal sources, but some of these are not in the endogenous locus, right? So I wanted more evidence from the endogenous locus. So in order to do that, we turned into this system, a CRISPR-I system, a CRISPR inhibition system, which basically works pretty simply. You use a guide RNA to bring this dCas9, the inactive Cas9 that is attached to a crab domain to this region, and basically it creates this steric hindrance and basically inhibits transcription. So if you have the guide RNA on top of the SNP, right, then you can look at the genes in the nearby region and show whether this element, at least this region that contains the SNP in the endogenous locus, whether it's really affecting gene expression or not. So for that SNP that I showed you, the RS165 SNP that is associated with this SNHG18 gene, we were able to show that indeed, when we have the guide RNA that's sitting on top of the SNP, we get lower expression of this SNH18, suggesting that this is indeed a regulatory element that contains the SNP. And we're doing this actually for all these other SNPs, but the data, we don't have it yet. We're working to test essentially all of them. And it will be really interesting to see how many of them we can validate with this method. Okay, so this is one line of evidence from the endogenous locus. Of course, I wanted even more because we have done all these epigenetic assays, right? And we can look to see if there's any allelic imbalance in the epigenetic assays. It turns out that one of our donors, because we did these in 6 donors, is heterozygous for that SNP, this RS165 SNP. So we just counted the reads that map to the different alleles. And actually, we were able to show that, when you have the A allele, you get more read counts from this, this epigenetic mark, H3K27AC in this locus. Again, showing that there is a, allelic imbalance, and that allele, obviously, these different alleles might be affecting accessibility in this region. So A allele showed higher accessibility compared to the T allele. So we have this data. We can now go back to our EQTLs and actually look to see if this effect is something we observe in our EQTLs, right? And this is basically the gene expression level on the y-axis and the genotype on the x-axis in the 2 different conditions. And the T allele, which had low read counts in the accessible, in this epigenetic assay, had lower gene expression in both the proliferative and the quiescent conditions, essentially fitting with the idea that, this is a regulatory SNP that's affecting the expression of this gene, the SNHG18. So the next question became, what is SNHG18? Because this is a non-coding RNA, and essentially nothing was known in the literature, its role in atherosclerosis or vascular smooth muscle cells, etc. So we collaborated with Minna Kaikkonen at the University of Eastern Finland and analyzed single cell data from atherosclerotic lesions. These are from the carotid endarterectomies. And what we found was that this gene is actually expressed in the smooth muscle cell cluster in the atherosclerotic lesion. So that gave us confidence that, what we're finding is actually a smooth muscle cell specific phenomena. And this is basically the different smooth muscle cell, single cell RNA-seq clusters. So this SNHG genes, and again, I still frankly don't know much about them. If they are in the nucleus, they can play a regulatory role in terms of, gene transcription. So my postdoc, we cultured cells, and my postdoc actually looked to see with RNA-scope if this long non-coding RNA was only in the nucleus in various different conditions in vascular smooth muscle cells. But unfortunately, it is not. This is not showing up that great, but there is a little bit of accumulation, it seems like, in the nucleus, but it's also cytoplasmic. So, we really cannot say that, it is only localized to the nucleus and having this gene transcription role. But of course, we had measured all these phenotypes, the proliferation, migration, calcification kind of phenotypes. So we thought, okay, why not just look at correlations with this phenotype and see if this gene expression is correlated with any of the smooth muscle cell phenotypes. And only one of them was correlated with this expression level of this gene. This proliferation in the PDGF conditions was negatively correlated with the expression level of the gene in our 150 different donors. So we then silenced this gene in smooth muscle cells and looked at proliferation. And just we would expect from this negative correlation, we actually saw an increase in proliferation when we silenced the level of this gene. We silenced it to about 70% or so. So, this is the way we're thinking about, identifying the causal variants, the effector transcripts, and the individual genes. Of course, there's a lot more work to do. There's a lot more loci. And now the one thing that we're trying to establish in the lab is using these immortalized smooth muscle cells to do SNP editing with CRISPR, and then obviously looking at the gene expression nearby to actually show that, changing the allelic configuration affects the nearby gene expression in the endogenous locus. My postdoc, Red One, then was also interested in splicing. He thought maybe some of the smooth muscle cell loci, the coronary artery disease loci, are actually affecting splicing. And so he did this analysis called SQTL mapping, splicing quantitative trait locus mapping. And in this case, basically what you're looking for is the effect of the genotype on transcript isoform abundance. You can see here, for example, in the presence of the C allele, you have a lot more of the short isoform compared to the long isoform. And in A, it's just reversed, right? It's just the opposite. So when we did this, we found 4,400 SQTL genes. So these are SNPs, nearby genes that affect the splicing of the genes. And similar to the EQTL studies, we did a colocalization analysis with the coronary artery disease loci. When we did the colocalization analysis, we found that there were 164 smooth muscle cell genes whose splicing was associated with the SNPs in the locus. This was a really surprising result to us because we are finding fewer SQTLs, but many more, almost twice as many coronary artery disease loci are actually colocalized with these SQTLs, which, if true, and I'm going to repeat this, if true, implies that majority of the molecular mechanism of this, disease risk is actually through affecting splicing as opposed to overall gene expression abundance, right? Which was kind of an interesting finding. But the reason I said if true is because we're using short-read RNA sequencing to quantify the isoforms. And, I know there are, really, advanced methods to do this quantification, but, imagine a case where there's a gene with 3 different isoforms. These are all the different exons, right? So if you have this short read, that's coming from your Illumina sequencer that's about, 300 base pairs long, it can map to any one of these SNPs. So you don't really know if this read is actually coming from this isoform or the second one or the third one. In order to really quantify this, you need reads that actually span exon-exon junctions, and they are rare. The exon-exon junction reads are not that many. You need to do really deep sequencing to have a lot more reliability in your isoform quantification. So we thought, perhaps a bit naively, we said, why don't we do long read sequencing? And at least if we do long read sequencing, which can, this sequencing up to 3,000, 4,000, 5,000 base pairs, we will at least be able to identify the isoforms that are expressed in smooth muscle cells. So if only one of the isoforms of this gene is expressed in smooth muscle cells, then we know that's our search space. If 2 of them are expressed, that's our search space, right? So we did a very similar experiment. We took 6 donors, 3 females, 3 males, and then we grew them in these 2 different conditions, the quiescent and proliferative conditions. And we collaborated, my PhD student Noah collaborated with a colleague, Gloria Shankman, who actually did her PhD here at University of Wisconsin in a proteomics lab, and to do long read sequencing. And we identified the expression of 21,000 genes and 81,000 isoforms. So that kind of became our essentially search space. But then the next thing we thought about was, how are the variants, the coronary artery disease-associated variants affecting splicing? And there are probably many ways, right? But we said, let's actually focus on the low-hanging fruit, which is basically the variants that are in splice acceptor sites. When the spliceosome is, splicing the primary RNA to make the mRNA, it recognizes these GU donor sites and the AG acceptor sites to make the mRNA. And if there are SNPs in these two nucleotides, then we should probably see a clear effect, right? And obviously there are not too many of them, there are few of them, and among the coronary artery disease-associated loci, but we thought that this might be one way to actually try to answer this question of CAD-associated SNPs and gene splicing. I'm going to show you one example from this. So this is a gene called PARP12. Based on our long read data, there are two isoforms that are expressed in vascular smooth muscle cells. There are actually many more isoforms known about this gene, but we can only detect the expression of two of them. And there is a SNP that is associated with disease that is in the donor acceptor site of this one small exon called exon 11, okay? And it either creates or disrupts a splice acceptor site. And when it disrupts the splice acceptor site, basically it either creates the long isoform that has the exon 11 or the short isoform that has the exon that doesn't have the exon 11. And when we look at the the transcript isoform abundance, you can see that there is a very strong effect where if you have the G allele, you're producing the long isoform. If you have the G allele, you are producing the short isoform, right? Because one of them is creating the splice acceptor site and the other allele is not. And the risk allele actually leads to the longer isoform than the non-risk allele. Again, I wanted additional evidence, so I asked Noah to actually culture smooth muscle cells with the different genotypes in the two different conditions. And so he took cells with the AA, GA, or GG genotype, and then he designed primers that, just PCR primers that obviously recognize these exon-exon junctions, and then looked at the abundance of the two different isoforms. So he could detect the expression of the two different isoforms, and you could also see that when you have the G allele with PCR, you actually don't have the short isoform, right? So only the long isoform is produced. So that kind of gave me again confidence that with this additional or orthogonal evidence that we might be onto something. These PARPs are ADP ribosylases, and again I know very little about this. They are primarily dysregulated in cancer. There are PARP inhibitors that are actually in the market for cancer. So we thought, okay, let's look at the 3D dimensional structure of this protein using AlphaFold, and see if there are any differences in the in the way the absence of this one exon may affect the enzyme. And it turns out that actually this exon 11 encodes this catalytic domain of the enzyme. So in one condition, you actually don't have the catalytic domain. In another condition, you have the catalytic domain. And what's interesting is that the risk allele is actually the one that leads to the full-length isoform where the catalytic domain is there. Of course, we haven't tested the function. These are predictions from 3D to AlphaFold structures. but it's a pretty, major impact if this is true, right? You might be impacting the function of this enzyme. And we have other examples like this. So for example, there is another locus that is affecting integrin, an integrin where the cytoplasmic domain is absent, for example, in the presence of one allele compared to the other. Okay, the final thing that I want to discuss is, a direction that the lab is going to understand mechanisms of sex differences in atherosclerosis. So there are many epidemiological studies that show that, women are protected from heart disease or coronary artery disease before menopause, and after menopause, they catch on. And if you look at pathologically, the plaques, there are actually differences even at the plaque level. For example, men tend to have more fibrous cap rupture when they have a myocardial infarction compared to women who have erosion of the fibrous cap, and that leads to a different type of pathology. But hormones don't explain these differences. I mean, there are certainly differences due to hormones, but they are certainly not the only differences. And there are additional differences that, are hormone independent. So we thought, let's, think about a genetic way, if we can answer it using human genetics. Sorry. There are certain loci, I think 10 now loci, that actually show a sex-biased effect in coronary disease risk. So I'm going to show you one of them. This is, again, one of these locus zoom plots of a locus that encodes this gene called TRF2IP. And this is when you combine both sexes, this is the association from the, with the CAD. But when you, look at only males or only females in this data set, despite having more females, the signal is not there. So the signal is actually coming from the males. So this is one of those examples of a sex-biased locus. And when we look at this locus, we do find this interesting phenomena where, the risk allele leads to lower expression of this TRF2IP gene in the nearby region in females, but it leads to higher expression in males. And in our at least phenotypic data, this is recapitulated in the calcification as well. So we're looking at SNP by sex interactions, and the risk allele leads to lower calcification in females, but higher calcification in males, which kind of fits maybe this process that we're observing with the G1s. And this is also like a really kind of interesting gene. We're still working on it. It can either have a role in telomere elongation or NF-kappa B signaling. So it can act actually as a transcription factor, or it's part of the telomeres complex. So it's pretty complicated. We don't know exactly what's going on. My grad student Lexi is working on this to kind of try to see if actually this locus is affecting obviously TRF2IP expression in the nucleus or cytoplasm, and whether it's having a role in P65 signaling or telomere elongation. But this is kind of the direction that we're going with this kind of studies. Okay, so just to conclude, we have quite a few loci that are associated with expression in smooth muscle cells, and even more loci that are associated with splicing. I showed you 3 examples of SNHG18 and PARP12 and TRF2IP that have these regulatory splicing or sex-biased effects. And these are the kinds of things that we're interested in further following up to really nail down the mechanism, the causal variant, and what the gene is doing in smooth muscle cells. So with that, I would like to thank, of course, all of our collaborators and our funding sources. And of course, I also want to acknowledge the people in my lab. I cannot somehow show their pictures here. Hopefully they will show up. Of course, they did a lot of the work. I'm really surprised that the pictures are just not showing up. Well, I would like to thank the lab. Thank you very much. Oh, happy to answer any questions.
I'll start with the question. So one of the most exciting things that you said was that there were more splice QTLs associated with coronary artery disease than there were expressing QTL, which is really a dramatic result. I don't think anyone ever knew that before. Do you see examples where the splice creates a new function?
We predict, I mean, obviously this PARP12, the enzymatic action versus non-enzymatic action is one example. The other example I mentioned was this integrin, which may or may not play a role. So there's a splice variant due to the coronary disease risk allele does not have a the cytoplasmic domain of this integrin. And who knows if it's playing a role in mechanism signaling or extracellular matrix interaction or something. There are actually a few more that I'm now, so Noah is focusing on like 7 of these because he predicted that the different isoforms would have a functional outcome, basically, similar to the PARP12 study. But what I really want first to do is to see if actually these protein isoforms are expressed in the cell. So we know that transcripts are expressed. So with Gloria, we're doing proteogenomics and targeted proteogenomics to figure out basically first if these different isoforms have been translated into proteins. There are some predictions for long non-coding RNAs too, but we are not even focusing on those because we have no idea if the different isoforms are having a functional impact. Yes.
Let's go.
Thank you.
Do you have an idea of some of the factors that will be affecting or causing those different genetic expression of those?
I actually had a hard time hearing.
Oh, okay. So some speculating what factors be involved in causing that gender.
Oh, that is such a good question. Yes. Yes, I see. That is such a great question. Yes. Okay, I guess I was supposed to repeat these questions for the Zoom audience. I'm sorry, I just now realized that. So what is the cause of, I guess, the sex difference? We don't know. TRF2IP, we haven't looked to see if actually hormones affect its expression, but what we looked at was if the SNP that we predict to be causal in that locus is in an estrogen receptor alpha binding site or not. And it's not, but there's a very nearby binding site. So who knows how it's affecting, if it's a cofactor of another factor that's regulating its expression and things like that. So we don't know the answer. That's one possibility that, the SNPs are affecting estrogen receptor alpha binding or activity somehow. Yeah. Yes. Thank you.
I was just wondering if the splicing issue you're potentially seeing is cell type specific or the impact just affects certain cell types more than others, and if so, how?
Yeah. I don't know if you know this, there's this GTEx consortium that looked at 40 plus tissues from about I think 900 humans, and they did this SQTL mapping. Theirs is a little bit actually less reliable because they only did single end sequencing and they didn't prepare the RNA in a stranded fashion, so there's all kinds of caveats with that data. But they did show that SQTLs in general were more cell type specific than EQTLs, although many I shouldn't say cell type, but tissue type specific, although many SQTLs were shared across the tissues. I have no clue how they are becoming tissue specific. I will speculate one thing though. We looked at SNPs that are in donor acceptor sites, but there are studies that show that these SQTL SNPs are enriched in regions of the transcripts where RNA binding proteins are predicted to bind. And some RNA binding proteins may have a tissue or cell type specific expression or function, and that may be the reason why we're seeing this, or they are seeing this cell type specificity in SQTLs. Very insightful question. I wish I knew the answer though. Yes.
I have a few questions. If you'll let me.
Sure, please go ahead.
So my first one is about the splicing QTL versus EQTL.
Mhm.
Do you see a difference in effect size for the disease between these two different classes? You might expect the spliced QTL to have one for the disease.
I think it depends. So again, I guess I skipped a lot of things. One thing some of that, there are 164 loci, that is SQTLs in coronary disease, and 85 that are EQTLs. Some of them overlap. So we actually looked at only the ones that are just SQTLs, because we weren't sure like if there's some bioinformatic analysis that we're not doing well and we're not getting, the SQTL, EQTL business becomes complicated, right? So everything that I showed is PARP12, the other integrin, etc. In those loci, we only observe SQTLs. We don't observe EQTLs. For the common ones, we did not compare the effect sizes, but honestly, like thinking about the plots that my students show me in terms of like genotype, gene expression, you're right. The SQTLs seem to be more distinct, more different between the genotypes, but we haven't done any kind of formal analysis. I think that's a really good idea.
My second question is about your cellular assays.
Mm-hmm.
So, those are much more simple
Mhm.
readouts compared to gene expression. Molecular treatments. Could you do GWAS on those?
We did. Yeah, we already published those. And so that's why I didn't talk about them. I would actually argue that they are not simpler. Because, in my mind at least, I think cellular phenotype, like migration, is a much more complex phenotype than, gene expression in a given locus, which itself is a complex phenotype, right? So we did GWAS, and we found that 79, I think, of the CAD loci are actually associated with with one of the phenotypes. Actually, I'm not gonna go. Yeah, this is the result, but it's in a paper basically. And what was really interesting about this was that this is again colored based on the risk allele. There are some loci that are only associated with calcification, some only associated with proliferation, some in opposite directions where the risk allele increases proliferation, decreases migration. So it was really complicated actually.
Yeah, simpler. I mean, this would be simpler reading. You get one number per cell line, and
Oh, I see. That kind of simpler. Okay.
yeah, and
Yeah.
yeah, and then the next question is about obviously getting healthy heart tissue. People are not donating that.
No.
So maybe can you comment a little bit about your samples?
Yeah.
And what maybe it would look like if you were to develop controls. Because these are all heart.
No, actually, these are healthy controls.
Oh, yeah.
So these are people whose hearts were deemed healthy enough to be transplanted. So we didn't take the transplanted heart. We actually take the vessel from the donor.
Oh, gotcha.
So, and these are people who mostly died early, in car accidents and stuff. Yeah. And you're right. Like we cannot biopsy, obviously, living people, for their arterial smooth muscle cells. So this was the best resource that we could find. Yes.
So, wonderful talk. I think after hearing that, maybe we should have had a different conversation earlier on some of these.
That's fine.
Some of these different things. But one question I had was, you have the one example where you went back then into the single cell data from the pack. And so that correlated very well with your smooth muscle phenotype. Are there examples that you have of that touch more of those cells involved in the plaque, or do you just discard that in your data and really only focus on those that might only touch the smooth muscle cells? I think each of those pools could be interesting.
Right.
Targets.
Right. So that's another excellent, very insightful question. So for just validation purposes, when we want to, what I haven't shown also is that we did immunofluorescence on certain other genes in coronary arteries from humans with different plaque compositions and things like that. Frankly, for those, we picked the genes that are smooth muscle cell specific, because obviously then otherwise it complicates everything, right? There are examples in our paper where the EQTL is smooth muscle cell specific. There are examples where the EQTL is smooth muscle cell specific and expression is in other genes, but there's no EQTL in those genes. In the cells, but there's no EQTL in those cells. And we had other examples of those where we see the expression of a gene in a adipose tissue, but the EQTL is only adipose and not in liver, blah blah. So there are quite a few of those examples, and we went through all of them in a paper that we published, basically. Yeah. And those are most likely being regulated in like cell type specific enhancers and transcription factors and things like that. Yeah. But frankly, many of these genes, like Randuan almost always picks like the gene where there's nothing on PubMed, and then makes wants to figure out at least contribute something to the field, and we make sure that they're expressed in smooth muscle cells primarily and things like that, just frankly to make our lives easier. Be practical a bit. Brian.
I was thinking about pleiotrope.
Yes.
So you're distilling down these potentially causal SNPs.
Mhm.
Obviously, there's LD issues, but you're getting closer to potentially atherosclerosis specific SNPs. Do you see that these SNPs are not pleiotropic without achievements?
Yes, there are some that are pleiotropic with hypertension. So the paper that we published on the secreted proteins, we looked at PQTLs, and we found some loci that are coronary artery disease, migraine. One of the SNP gene pairs that we found, we narrowed down to 18, is also associated with lipids, for example. We don't know if that is, just fell through the cracks, or if it's, truly associated with lipids and also vascular smooth muscle cells. So there are definitely some.
Do you think that these SNPs will help improve polygenic risk?
I think so. Yeah. I mean, I talked to a lot of people, about this, of course, because, the annual meeting of the American Society of Human Genetics, half of it is PRSs, basically. Yeah, maybe I'm thinking too simplistically. So, there are other geneticists in the room, obviously, but, when you're looking for a Mendelian disease, right, you're genotyping a certain variant or known variants, and I really think in a very maybe simplistic way that way, right? There may be certainly more than one causal variant in a locus, but the polygenic risk scores, the one, for example, that I guess Katharaysan had published, for example, is looking at all the variants that were, assayed, millions, and there's a lot of noise in there, right? I mean, it's a hunch. I don't have proof, but in maybe 10 years. But I would honestly love to hear people's ideas about that. It's just a thought.
Okay, I think you saturated everyone. Thank
Well,
you.
the students are saving their questions for the
That's-
Thank you, everyone. Thank you so much.